Imagine if we could pinpoint exactly where on our vast DNA that proteins love to hang out. That's exactly what ChIP-seq lets us do. Let's dive in and discover how we can map these molecular meetups. Hey guys, welcome back to Bytesized Bioinformatics. Today we are unraveling the mysteries of ChIP-seq and discovering the dance between proteins and DNA. DNA isn't just a passive string of letters. It's constantly interacting with proteins which regulate its function. Knowing where a protein binds to DNA is crucial. It can tell us how genes are turned on or off, how cells respond to environmental cues, and even why certain diseases develop. But here's the challenge. With billions of base pairs in the human genome, how do we know where specific proteins bind? ChIP-seq comes to the rescue here. It's a technique that combines chromatin immunoprecipitation, or ChIP, with sequencing. Here's how it works. First, we start with cells where proteins are naturally binding to DNA. We use a chemical to freeze these interactions. This is often referred to as cross-linking. It's like capturing a moment in a picture, ensuring that everyone stays in place. Once our interactions are stabilized, we fragment the DNA into smaller, more digestible pieces. This ensures we can analyze them efficiently. Next, we use antibodies that are specific to our protein of interest. These antibodies bind to the protein, allowing us to selectively pull out the protein DNA complexes from the mixture. It's like going fishing, where each protein could be caught with a specific antibody hook. The DNA fragments that were bound to the protein are now prepared for sequencing. We first separate the protein and DNA, and then amplify the DNA so we have enough. After sequencing, we can see the precise DNA code where our protein was interacting. Once we have this sequencing data, we can now pivot into the realm of bioinformatics. By aligning these sequences back to the reference genome, we pinpoint the exact locations where our protein was binding. These binding sites manifest as peaks in our data, showing the protein-DNA interaction hotspots. Delving deeper, we can analyze these peaks to find patterns, understand gene regulation, and even predict potential functions of the protein in question. ChIP-seq is a revolutionary tool in genomics. It provides a high-resolution map to DNA-protein interaction, and this can help us understand gene regulation, find enhancers and silencers, and even shed light on where these interactions go awry. By knowing where proteins interact with DNA, we get a clearer picture of the complex choreography that keeps our cells functioning. So that's it for our quick dive into ChIP-seq. I hope you enjoyed the episode. If you did, leave a like, and I'll see you in the next episode.